Thanks for the introduction. My name is Juliana Chavez, and today I'm going to present patient cell models of childhood and aging dementia to investigate drug repurposing and drug delivery. So I tried to put a bit of introduction because I knew some parents would be here. Oh, okay. I'll try that one. So just a little bit of background. So dementia is known to be a broad term to describe any disease that has decline on. It's not working. Okay, sorry. Is it working? Yeah, no. Dementia is a broad term used to describe a decline in cognitive function, which includes but not limited to memory, language, confusion, and learning. We know that dementia can be subclass in different two types. Aging dementia, which typically occurs on older adults, typically older than 60 years old, including Alzheimer's, Parkinson, and the motor neurodegenerative disease. And we also know that childhood dementia is another type of dementia that could normally account for kids around 2 and 5 years old. So we know that childhood dementia is rare, but there is more than 145 genetic childhood dementia conditions, and in every one in 2,900 babies born in Australia are currently untreated with childhood dementia. So 50% of these kids will die by the age of 10, and 70% will not live past 18. So even though childhood dementia like kids die with childhood dementia is the same number as childhood cancer, the amount of research funding available per patient is almost 43 times less. And because of that, there is not many treatments available. So we need more funding to look into the rare disease. So for the purpose of this presentation today, I'm going to focus on Batten disease, which is the most common childhood dementia type. So we know that symptoms are included but not limited to vision loss, muscle weakness, seizures, loss of coordination, and cognitive impairment. There are 13 types of Batten disease already described in the literature, but today I'm going to focus on the CN3 juvenile Batten disease, which is the most common Batten disease type. So we know that Batten disease, CN3 juvenile Batten disease, is caused by a mutation in CN3 gene, and the symptoms usually begins between the age of 5 and 10. There is no treatment available for Batten disease, CN3. Most of the treatments available are to manage the symptoms. So we know the pathology of the Batten disease. It's in, as you can see in the figure, in a health patient, the lysosome works normal, but in Batten disease, because of the mutation, the CN3 gene, there is accumulation of toxic and undegraded waste material, which is called lipofuscin. So because of this lipofuscin, the accumulation of this undegraded waste material, leads to inflammation and oxidative stress, which could lead to disruption of neurons and other brain cells, such as astrocytes and neurons, which leads to the neurodegeneration and um, and and the kids will like without the treatment can die and start like the cognitive decline. It's really rapid. So as I said, there is no many treatments available for Batten disease. The only type of Batten disease that actually has treatment available is CN2. However, the annual cost exceeds almost $500,000 per patient per year. And it received approval in Australia in 2018. And when administered early, it can extend the life up to 20 years. However, without this treatment, children with CN2 Batten disease will die between the 6 to 12 years after diagnosis. In order to try to come out with a solution for to find some extra, like more treatments for Batten disease and rare disease, drug we're proposing has a great potential to come with a solution. As a part of strategy to intensify effective treatments for Batten disease, this project, together with help with Dr. Zachary, we use a drug we're proposing screening for extending existing compounds that to uncover different potential for these compounds that has already been available. Just to briefly explain and try to be as simple as possible, we know that some approved drugs that has already been approved for different diseases, such as COVID-19, could has different purpose if we investigate it for a different disease. So there is a computation analysis where you can get some data and from all the compounds that has already been approved, and you know what's the mechanism that these compounds, how it work. So it actually reduce the development timeline and lower the development cost, because when we start to looking for new drugs, it takes time to find approval for safety and to test the interactions with other organs and so on. So it could reduce that because it's already been approved, reduce the risk of failure, as we already know the mechanisms of these drugs. And the treatments could be an option for rare disease where there is no many fundings available. And could be a better use of existing drugs. So what Zach did when he was trying to using drug we're proposing to investigate potential Batten disease drugs, he used the risk genes that we know are already being identified for that are the mutations for the Batten disease, such as PPT1 or CN3 gene. So then he investigated how these genes could co-regulate in the gene network, and what does these compounds that he could find on the data analysis that would interact with the gene, like whether it modulated the gene or inhibited the gene. So after this really fancy analysis that he managed to have done, he found approximately 80 compounds of that could be potential for treating Batten disease. And we have choose the top 5 that has the best affinity. So after doing that and knowing the compounds that we would like to investigate on our project, we used human immortalized neuroprogenitor cells, which is pretty much a commercialized cell line that we can easily grow and differentiate it into brain cells, such as neurons, as you can see in this image. And red is a neuron after 14 days of differentiation. So we tested out the drugs in different concentrations to see if it could affect the cell, the quality of the cell. If the concentration that we have choose could induce to cell death, for example. As you can see here, there is no significant change compared to untreated, which suggests no cytotoxic effects of the chosen drugs were observed in any of the concentrations used. So to move on, and also to be able to use model that we could investigate the disease mechanisms of Batten disease or any other childhood dementia, we have used induced pluripotent stem cells that has many advantages. As you can see here, we can collect the samples from health patients and patients with the mutation. And then these cells, after reprogrammed, we can create it differentiated then in different brain cells, such as astrocytes, neurons, microglia. So why this model is good is because we can do a patient-specific modeling where we can compare healthy versus the disease one to see what is the difference between them. Um, we can access to the affected cell types. In the case of Batten disease, it starts with neurons and astrocytes, where there is accumulation of the lysosome lipofuscin on those cells. And then we can use as a drug testing. So as I said earlier, we have identified 5 potential drugs that has been already in the market that we could see if could does any effects on trying to treat the Batten disease. There is also a possibility of gene editing compatibility, which we could correct the mutation and see if we fix the mutation, if we could reverse compared to a health control. So after selecting the modeling that we would use to investigate this disease, we then use the cells that we got from Professor Tony Cook from University of Tasmania, and we differentiated those cells in cortical neurons and astrocytes. As you can see here, we have, we have the CN3 Batten disease in the top here, and also astrocytes, and also neurons and astrocytes from health donors. And after differentiating into those cells, we investigated the lysosome markers in corrected one compared to the CN3. And as you can see here, we have significant, it's upregulated in CN3 corrected, where the mutation was corrected compared to the mutated one. And then later on, we also investigated the inflammatory response in astrocytes by cytokine secretion. As you can see here, in the corrected one, we had a lower secretion of these inflammatory markers compared to the Batten disease patients for 2 of the 3 markers that we have investigated. So later on, we also wanted to see if the drugs that we have selected could actually induce the lysosome expression in CN3 neurons. As you can see here, from more than one drugs, we could found a higher expression of these lysosome markers compared to the untreated one, which suggests that treatment with these drugs candidates upregulated the lysosome enzyme expression in CN3 neurons, which could be a potential treatment. So then moving on, we also investigated if these drugs treatment could decrease the inflammatory response in astrocytes from these Batten disease patients. And as you can see here, the drug 3 show a really reduction in inflammation based on our cytokine secretion, and which suggesting that the drug might alleviate the inflammatory response in the CN3 astrocytes. So another thing that we also wanted to investigate after treating these CN3 Batten disease astrocytes, remember that I said in one of the diseasing signs is like accumulation of the lipofuscin due to the mutated Batten disease. As you can see here in green, this is the accumulation of the undegraded waste material. And only after 24 hours, we could see a clearly reduction of the accumulation of these undegraded and toxic material, which suggests that only 24 hours treatment, lipofuscin accumulation was significantly reduced in CN3 astrocytes, and indicating that the drug potential to clear toxic material in Batten disease cells. So, in summary, for this part of the project, we wanted to highlight that computational approaches can link drug and gene interaction data that could be a promising identify novel candidates drugs to treat rare disease. It is critical to explore drug repurposing for rare disease to reduce the expensive and the time-consuming drug development. Batten disease, iPS-derived brain cells, neurons, and astrocytes demonstrated a significant demonstrate as disease phenotype, as you could saw with the accumulation of the lipofuscin on the astrocytes from CN3 Batten disease, which allows us for testing the effects of the drugs and highlighting the translated to patients. The drug candidates promising effects of the lysozyme gene expression, inflammation, and the lipofuscin accumulation. But only after that, we also wondered to see one of the big challenges for treating neurological disorders is the drug delivered due to the blood-brain barrier, as Professor Anthony and Dr. Yahoo, I'm sorry about mispronouncing your name, already said. So we also wanted to use our iPS cells to derive a model that we could investigate the brain endothelial cells, which is the most common cells for the blood-brain barrier. So, in my PhD, I have investigated the FOXO treatment as a way that we could improve drug delivery and therapeutic response in Alzheimer's, and we now wanted to do with the Batten disease and other childhood dementia disease. So, just to give you a more explanation why the blood-brain barrier is important. So we know that the blood-brain barrier is composed by many brain cells, including astrocytes, neurons, pericytes, and microglia and the brain endothelial cells. The main function of the brain and the blood-brain barrier is to maintain the homeostasis. By regulating the permeability, what's going in and what goes out, that's how it regulates. But due to that function, a lot of drugs cannot penetrate into the brain due to the size or because the blood-brain barrier would not allow it to penetrate. Because it protects the brain from toxins, pathogens, and harmful molecules. However, when disrupted, the blood-brain barrier might allow harmful substances to enter and reduce nutrient supply and trigger inflammation, which we know is not good for any neurodegenerative disorders. So we know that blood-brain barrier damage is linked to neurological disorders, including Alzheimer's disease, Batten disease, and others as well. So what we did during my PhD and in Alzheimer's disease, we used Fox ultrasound treatment to open the blood-brain barrier to improve drug delivery. This wasn't my idea. This has been already established in the field. So pretty much what we have done, we have used the iPS-derived cells to create brain endothelial cells, as you can see here in green and in red. And then we expose the cells using Fox ultrasound. And it's been shown in animal models and also in vitro models that after adding the microbubbles, and once we induce, we could open the blood-brain barrier without breaking the blood-brain barrier, which could be a non-invasive way to temporarily open the blood-brain barrier up to 24 hours to enhance the drug delivery into the brain. So previous studies has investigated the effects of the Fox ultrasound to treat neurological disorders. And as you can see in some of them, it shows clearance in amyloid beta, which is accumulation of some proteins in Alzheimer, and also improve memory in vivo in animal models. And it has been shown to improve the drug delivery through the blood-brain barrier. And more recently, it has been shown to also be used to as a treatment to reduce the tremors in Parkinson's disease. So the reason why I brought this is just to show you all the possibility of the using of Fox ultrasound to treat to treat neurological disorders. So during my PhD, I was part of this really amazing study that we have done, where we investigated if we could increase the permeability in the brain. As you can see here, we have significant increase the permeability was observed after following Fox ultrasound treatment for the two phenotypes that we have investigated at the time. And we also tried to see, we tested some drugs that has already been approved for Alzheimer's disease to see if we could improve the drug delivery. And as you can see in these results, we saw really increase in the delivery of this antibody that has been used in the therapy of Alzheimer's, and which could show again the possibility of using the Fox ultrasound treatments to improve the drug delivery into the brain. So as my take-home message for you today, I have talking about Batten disease, Alzheimer, Parkinson. So we want to now investigate the Fox ultrasound treatment to enhance the drug delivery into the brain by using the 5 repurposing drugs that we have already selected to see if we could see a better outcome after Fox ultrasound treatment. We want to combine these two ways to treat Batten disease. And based on our results, we show promising potential of Fox ultrasound and drug repurposing as affordable, non-invasive therapeutics for rare disease. And iPS-derived brain cells combined with drug repurposing and Fox ultrasound give us a powerful platform to study patient-specific treatment response. And this strategy supports the development of personalized therapies for both childhood and adult neurological disorders. And overall, our work opens avenues for target, cost-effective treatments for conditions with limited options like Batten disease and also NPC disease. I would like to acknowledge my team and our collaboration. And if you have any questions, I'm open for. And if you'd like to collaborate or even connect to later on, message me through LinkedIn or ResearchGate. I'm more than happy.
Do we have any questions? Yes, we do. Yes.
Hi, that's really loud. Thank you so much for your talk. I actually had a bit of a question. I'm not sure if it was addressed and I just missed it, so I'm very sorry if it is. With the focused ultrasound, you said that it disrupts the blood-brain barrier for 24 hours. Have you looked at any potential adverse side effects of that, such as like increased neuroinflammation after the disruption of the blood-brain barrier?
Yeah. So it does not disrupt it. It does not break it. Like the blood-brain barrier, it's really tight because of some of the time junctions proteins. So because it's really tight together like that, Fox ultrasound, when combined with microbubbles, it kind of like moves around these time junctions, which leave a little bit more space. Does not break, which does not lead to increased inflammation. So we did have check with just Fox ultrasound, with Fox ultrasound plus microbubble at immediately in 24 hours. It does not increase the inflammation.